Okay, so we have looked at the important features and properties of DNA. So remember, we're going from that chapter 2 handout on the central dogma of molecular biology. And we've covered this part of predicting how scientists use the features and properties of DNA for discovery, because we've looked at the structure of DNA. Now, the next question, it looks at how the DNA fits into the bigger central dogma of biology. And full disclosure, I really hate the term central dogma of molecular biology. I was a psychology major as an undergrad, so I wasn't immersed in this science jargon. And when I went to graduate school, they automatically just started talking about the central dogma of biology. And I was like, I don't have any idea what you were all talking about. That's gibberish to me. And it wasn't until they said DNA to RNA to I was like, oh, I get that. But they were just using a term that I hadn't known before because I didn't have this type of class as an undergrad. So just when we go and talk about the central dogma of molecular biology, just recognize that we are going to use that as our big picture for this entire course. We're going to zoom in on different parts of it, and I still don't like the term. All right, so what we're going to do is you can draw this on your handout, and then what I'm going to have you do when you come to class is just start with a blank piece of paper and try to do it again. And then guess what I'm going to have you do on the exam? You're right, draw it again. And we will add things to it as we go throughout the semester. So we can start here with just this blank spot for your handout. And then you also have a resource in our full PowerPoint slide that outlines it all ready for you, but in a drawing type of a way. And if you like to draw things and this works for you, you can go in and draw the specific details. Or you can just go with the names. So when we are talking about the central dogma of biology, we are really just taking this right here. If you can think about what this is, it's double-stranded. It's got a 5' and a 3' that's lined up anti-parallel for those two different strands. And so, and you see A, T, G, and C within there. So hopefully you're recognizing that in this central dogma, we are starting with something we already know. We're starting with that DNA. So when we're talking about getting it started, we are starting with DNA. And then this little circle right here, when you have DNA and you make more DNA, that is a specific process. I generally just do it like this. It's just a little circle. So you've got a section that we're going to talk about in class that focuses just on this. And then we have another process. So all of the arrows are processes. And we go from DNA to, and this one, I'm going to represent it a little bit different because we will add to this later in the semester. And I want to do it now and set you up for knowing what we're adding. So basically, that one says DNA to mRNA, but there is a step where you have regular RNA, and what we'll learn in a whole chapter or a portion of a large portion of a chapter is that there are bits of RNA that never go through this other process here to give us mRNA. So I want to highlight that it's not just DNA and automatically finishes with mRNA. There are other bits of RNA that get created and play an important role in genetics and biology. And when they first came up with the central dogma of RNA or of biology, it was just DNA, mRNA, protein. That's what's important, and that's all that matters. But in science, it's always more complicated. And now that we know that it's more complicated, I'm adding this in here. And then hopefully I have enough room to write everything and you can see it. But so basically we're starting with DNA. We are generating RNA, then changing that a little bit to mRNA. And then our final product is a protein. And then that protein or the expression of that protein or the regulation of that protein is going to give us a specific phenotype. So those are the bits. Relatively simple. If you can map this out right now, like I would recommend starting with a blank sheet of paper and they go, okay, DNA, RNA, mRNA, protein. Yay. Oh, and phenotype. Now, we're going to label what these things are. So if you think about DNA and making more DNA, we're just replicating it. We're just making copies of the same thing. So that is replication. And here's one of the areas where students actually know more about this than they think they do. When you think about, well, when would your cells replicate? And students are like, well, I don't know. And then we say, but if you're gonna replicate your DNA, why would you need to replicate that DNA? And then eventually we get to, oh, you're making another copy of the cell. And then we focus in and realize that you probably have had this in your high school biology courses and in your intro biology courses. Oh, right, we spent a lot of time on mitosis and meiosis, right? When cells are undergoing mitosis or meiosis, they need to make a copy of that DNA, and that is the whole replication process. And as you can imagine, in genetics, we're going to zoom in on this and talk about mitosis and meiosis in more detail. And then when we go from DNA to RNA, we are writing in a similar language. There's slight differences, but we're basically just rewriting it. And if you rewrite something, we call that transcription. So we're going to transcribe at this second arrow here. Well, so remember that first arrow is the loop. And the first arrow, when you're going from DNA to RNA, that is called transcription. And we're just rewriting it. And then this one, we're changing the RNA a little bit to make it messenger RNA. And in that process, we just call that RNA modification. So really, if you think about the terms, you can kind of figure out where they go, right? So to transcribe something is just to write it a little bit differently. To take one thing and convert it just a little bit, cut some things out, add some things onto it, you're just modifying it. And then this next arrow here, when we're going from that mRNA to a protein, if you think about, we know that DNA and RNA consist of nucleotides. I want you to think about, do you yet know what a protein is made up of? I want you to think if you can come up with that term. What is a protein made up of? And hopefully maybe it's a little bit of a read from those intro bios where the proteins are made up of amino acids. Another term for amino acids is peptides. So if those are familiar to you, the amino acids, also known as peptides, are what make up proteins. So basically, we are taking this language of RNA and we're changing it into a completely different language. We're using it as a code to change it completely. So if you think about changing from one language into a completely different language, if you think about that term, that's a translation. So we have transcription when we're going from similar things, and we have translation when we're just completely changing it to something else. We're using completely different, sometimes letters, and we're using completely different words. We're translating it into a different product. So those are all the points for the central dogma. If you can now cover this up, get a blank piece of paper and redraw this or rewrite this in a way that works for you, then you are well on your way to having this section of it. And remember, this is the big picture for really our entire class. And we're going to zoom in on replication when we talk about mitosis and meiosis. There's a whole chapter on transcription. There's a whole chapter on translation. There's a lot of how the protein leads to the phenotype, or expression of different proteins lead to changes in phenotype. And we even have a whole like big section on different types of RNA and how they are going to function. And what we will do in the next couple of classes is this doesn't just happen randomly, right? There are specific other proteins in the cell that are going to regulate how these things happen. And so we will also add on to this what kind of enzymes are driving each of these steps. And back to our big picture of the techniques or how we use the properties of DNA in scientific discovery, we will look at how are we going to use these steps and use these enzymes to make it work for scientific experiments. And if you're following along in the PowerPoint, like I said, we'll start with a blank piece of paper during class as well. And then I have a card sorting exercise where you're looking at the regular non-disease state of how this happens. And then we'll also look at a disease state, specifically sickle cell disease. And we'll think about how changes in DNA are going to lead to changes in our mRNA, which will then lead to changes in our protein, which will then give us a slightly different phenotype.